It's your host, Paige Jarreau, and you're listening to the FaceTime with Scientists podcast. Today, we're going to be talking to Isabelle Neylan. She's a postdoctoral researcher at LSU studying copepods. Isabelle, you are a postdoctoral scholar and researcher at LSU, studying marine invertebrates and how they respond to stress. So I got to
Yes.
speak with you and do a little video a couple weeks ago, so I'm super excited to chat with you today. So you're in the lab now. What are you about to show us, or what are you doing today?
Yeah, it's good to see you again, Paige. Welcome back. Today I'm working on RNA extractions. So we have copepods from the lab that we've been working with, and we take about 50 to 60 of them and we grind them up with kind of like a small immersion blender. You make a copepod smoothie, is what we call it. And then we put some chemicals in. It's called trizol. It's this bright pink compound that just helps all that tissue break down. And then we, did those first steps. And now all of our samples are in a centrifuge, and they just finished a moment ago. So perfect timing. And so from there, we're going to start extracting RNA. And I'm happy to walk through any of the steps I'm doing while I'm doing them. But really, it boils down to we're just going to pull the RNA from this mixture that we made, and you'll see it hopefully really clearly. This is the part I love. It turns different colors. Basically, it's like a rainbow connection moment. And then we're just washing the product. So we're going to pull out the RNA, and then we're just going to wash it with a series of buffers. I have them laid out here, and they're going to go through these filter steps. So this is a kit that scientists use all the time. So all the steps you're seeing are really common. Anyone who works with RNA, it's not specific to marine invertebrates. And so, yeah, I'll just be washing RNA while we chat. That's kind of where you've caught me in this
Awesome.
moment.
Amazing. So yeah, we've chatted a little bit and feel free to kind of get started and show us some stuff and
Perfect.
talk through it.
Let me
But
just pull
I
them in.
met with you in terms of learning a little bit more about copepods. I didn't know
Mhm.
you mentioned that SpongeBob has a friend who the little
Yeah.
green guy is a copepod for people who don't
Yes,
know. So
plankton.
can you talk a little bit about what a copepod is and why you study them?
Totally. Yeah, so they are small crustaceans, so related to things like crabs and shrimp and lobsters, but actually quite diverged if you look at their family tree. So there's a lot arthropods, insects, they're all kind of part of that same phylum. It's really diverse. And they are in freshwater environments, but also marine environments. So the copepod we work with is a sea creature, and they typically are quite small, usually about 1 millimeter to 2. Our guys are about 1 millimeter long, so visible to the naked eye but small. So if you think about SpongeBob's friend Plankton, small things that are floating in the ocean, a lot of that plankton, we calculate up to 80% even of plankton we find in the ocean are members of the copepod family. So they're really important in marine food webs. So, the classic little fish eat copepods, bigger fish eat the little fish, and so on up the chain. And they're one of the largest sources of carbon in the world and one of the biggest animal groups by biomass. Even though they're quite small, there's a lot of them.
Yeah, that's super cool. Yeah, no,
Yeah.
thinking about, right, climate change and impacts of heat and stress on that base of that food web, you might not think about it. They're just kind of normally invisible to us in terms of
Yeah.
impact on the world and the ocean. But at the end of the day, right, like a collapsing food web would be very
Really bad.
catastrophic,
Yeah.
really bad.
Exactly. That's a perfect word.
Yeah. Well, so what are you doing? You're kind of pipetting, you're
Yes.
talking about extracting RNA. What is RNA? Like, why are you
Yeah.
extracting
So
it? And tell us a little bit, like, we
totally.
know about, probably most people have heard of DNA in terms of the blueprint of life and what
Yeah.
dictates how our bodies are
So
going to work. But yeah, what's
I'll give
RNA?
you like a brief little visual. So what we have pulling out here. So I mentioned that compound we put in as trizol is that bright pink at the bottom. So anything that's not DNA or RNA gets kind of pushed to the bottom and is part of that pink layer. And then it's a little hard to see, but there is this kind of film that's right in between the pink and the clear layer.
Right.
That's DNA. So you can, in theory, I don't know if I have the finesse and the skills to do this personally, but you can, in theory, extract DNA also from this tube right now. But the layer we're going to pull and work with is this clear layer on top. And that's all of our RNA. So as you're watching me pipette, I'm just very carefully pulling that clear layer without trying to disturb anything else and trying not to disturb the film that is DNA. And then that pink layer, that's all the tissue and everything else that didn't quite make it into the RNA or DNA piece. So you can think of the exoskeleton of a copepod like a crab shell. They have this hard exoskeleton. That's all kind of coming to the bottom. And we really are interested in RNA specifically. So as you mentioned, DNA is the building block, right? That's, the I like to think of it, if it's a book, those are the words on the page, right? Your DNA sequence dictates what's going on in the story. But RNA is what gets expressed and ultimately becomes the proteins that will allow you to do all the things your body needs to do, right, in the cell and beyond. And so gene expression, I like to think of the metaphor. So if DNA is a book, gene expression is what page are you on and what words are you actually reading. So all those words might be in the book, but it doesn't mean you're using them all at the same time. And so how you're using your DNA and which proteins you're making when tells us what's going on physiologically and at the cellular level in an organism. And in particular, when we're thinking about stress, we want to know, is it going haywire? Are there preventative actions that they can do? So it gives us a lot of mechanism into how they're coping or not when we are exposing these animals to stress.
That's
I
super
got one
cool.
more.
Yeah, and I have,
Yeah.
questions for you later in terms of
Yeah.
getting
Yeah.
into the weeds of like plasticity and what it means when
Yeah.
changes or the ability to change is kind of baked into our DNA versus when it's this ability of our DNA to kind of create different products through this process of creating RNA. But in terms of you mentioned right the layers separating, like how is
Yeah.
it's separating RNA and DNA? Is it different weights? Is it like
Yeah. Oh, that's a really good question. So I'll load these up in this is a centrifuge, so just a machine that we are going to put our tubes in many times, that spins them around really fast so everything pulls through. And like I mentioned, there's DNA and RNA in the sample. So this column that we're putting it in first actually will hold on to any DNA. And again, with these chemical reactions that someone, I wouldn't say smarter than me, but someone with a different skill set than me has figured out, there's like a specific chemistry for when does it get stuck to a filter, when does it flow through a filter, when does it get attached into that kind of clear layer, when does it go down into that film. And so this molecular work is kind of wild. I think of it, it's a little like magic, like you put together a bunch of clear liquids and then like data comes out of it. So it's a little mysterious to me how it works mechanistically, but it's a really good question. And really, I think anyone can do molecular work. I was told that when I started doing this, and I think it's true. If you can follow a recipe, which is really what I'm doing here, you can do this process. It's just what to add, how much, and when. And something about that specific order and the chemistry, whether it's an alcohol, is it a salty buffer? All of those different steps are going to either keep our RNA or flush it out when we need it. And then the filters are going to hold on to what we want. So that's what we're doing for the rest of the
That's
podcast.
awesome. No, that's awesome.
Yeah.
So, yeah, I think people are familiar, maybe with DNA, with the sequencing initiatives like the whole Human Genome Project, where we're
Yeah.
trying to figure out what this baseline like blueprint is to understand differences between humans and other species or mutations that might cause disease or that kind of thing.
Yeah.
In terms of looking at RNA, is that something that's more recent in terms of like RNA sequencing? And that, like, yeah, what are
Oh,
you
that's
specifically
interesting.
doing with the RNA? Are you sequencing it? How are you then determining like what genes are turned on or off based on? Yeah.
Yeah. I don't know about like the like history of the science between like DNA versus RNA. We've known about them for a long time, right? When we figured out the structure of DNA, it came along with this knowledge that that double helix that breaks apart is both what allows the DNA to replicate in our cells when they divide, right? That's the classic like Watson, Crick, Rosalind Franklin story. But that ability to kind of break in half and then replicate is also what RNA is doing, right? So it's going along a single strand of DNA and making its complementary pair and then pulling off that sequence. And then this is, the central dogma of
Yeah.
biology, like certain groups of those RNA sequences tell you what amino acid needs to get put on the chain, and then that chain becomes a protein. So I think, we've been working towards these kind of mechanisms for a long time. I think you just ask different questions when you're working with DNA versus RNA. And so again, when you're thinking about the words on a book and what's in the book and how are different books different and how have they evolved over longer time scales, you want to look at DNA. How are things adapting over multiple generations? And we are working with DNA and the copepods as well. We're doing a population comparison. So the same populations I work with and think about RNA, myself and other members of my lab are doing DNA analyses as well. So it's helpful to do both, like it's two sides of the same story. RNA tells you more about what's happening in the moment. So when I pull a copepod out of the tube, like let's say it's heat stressed or not, I'm going to get a different signature between that control copepod that's, lived a stress-free life versus the one that's coming right out of a hot water bath, for example. And I want to know what are those differences in that moment? How is it different? And then if I want to know between two populations, one that lives in a warmer place, one that lives in a cooler place over the long term, how are their genes changing? Then we can look at DNA. So it's all different pieces to the puzzle, but they complement each other.
Right. Well, yeah, so as I was learning more about your research and then kind
Yeah,
of looking over it again and, because this is, I mean, it's complicated
it's totally
science
complicated. Yes.
in terms
Yes.
of what you study, like the genetics of these changes. But I was really interested, and you were talking about there's different ways that an organism can evolve. It can evolve kind of
Yeah.
like slowly over, like you said, multiple generations through like
Yep.
changes in the underlying DNA,
Hmm.
but can also change through this like plasticity where
Yeah.
maybe even one copepod parent is stressed and maybe they adapt in some way, but not
Yeah.
the underlying genetic blueprint, but there's ways that they can even pass that on
Yeah.
to like just one generation down,
Yeah.
So can you tell us a little bit about that? Like, how are these like,
Yeah.
yeah, changes happening, these adaptations, what you call plasticity,
Yes.
that's not so long lived, that has more to do with RNA and gene expression?
Yeah. So I will say I've been like plasticity is the thing I'm interested in most. That's what I did all my dissertation work on. Even as an undergrad, I was interested in these questions. They've kind of been following me. Um, and for a long time, I looked at it at the phenotypic level. So I, was looking at how it changes in, for example, heat tolerance or survival. And you can measure plasticity in that way. It's just the ability of an organism to change in response to the environment. So they're not changing anything about their DNA. It's, within a generation. Oh my goodness, give me one moment. The
I
lights in
see, yeah,
here
you
go off
have
sometimes.
you have, like, those, automatically dimming lights, and it's
Yes.
which is great for energy
Oh,
efficiency, right? But maybe not so great when
yes.
you're sitting at the bench for a long period of time.
Exactly. They should be motion sensing, and I think sometimes they forget I'm here.
That's hilarious.
Yeah, this has been a recurring theme in my extraction journey.
Well, you're new to this, you're in a new building, right? A new lab, so I'm
Yes.
sure there's,
So
like, things you're learning in terms of, like, what works best and what doesn't in that new space.
yes. And we're all learning together, and yeah, we're in a communal lab space. We're all learning to, how do we work together? And everyone's been great, but it's been and fun to work next to labs that I wouldn't get to talk to otherwise. But yes, we are in the new integrated science building on the main campus. Up on the fourth floor is our new lab space. But one side effect is that the lights sometimes go off in the middle of an extraction.
Hey, it's ambiance.
What was I talking about? Plasticity. Um, so again, the very basic definition is just an organism's ability to change their phenotype. So some measurable trait in response to the environment. Why RNA links to that is because that's how you kind of get a mechanism behind those changes, right? So for example, in our copepods, they have a plastic ability. If you heat stress them just a little, you don't kill them, but you just kind of give them a little bit of a heat stroke and then give them a day to recover and then measure their heat tolerance. It actually increases. So they have this beneficial acclimation ability. We call it heat hardening. And so that's really neat. And, the story could end there, but we're really interested in what gives them that beneficial acclimation. What is the mechanism? And so what we find is that that's coming from the ability to upregulate or increase the expression of genes involved in that heat stress response. So they make things called heat shock proteins, peptidases, it's another group of proteins. And when they make it ahead of time and carry it forward to the next day, they're better equipped. They have what they need on hand. And so that tells us the mechanism. And then we can take that another step further, which is kind of work in progress at the moment. And we can look at are the genes that we see that are differently expressed in the RNA that we study, are those the same genes we see that are different across populations? So that adaptation over many, many generations. And what we found is that, yes, the genes that are more plastic, the ones that change in response to heat stress, are also the genes we tend to find that are different between populations that are more southern, for example, and more heat tolerant, and those in the north that are more heat sensitive. And so these environmentally responsive genes are also the genes that are causing these populations to diverge. And we see that at the phenotypic level, we see that in the RNA, and now we're starting to explore it at the DNA level and look at genomic comparisons.
That's
Yeah.
super cool. So yeah,
Yeah.
what I thought was interesting, and this is where the story gets a little bit complicated, was
Yeah.
that, and I was still trying to wrap my head around it after we talked for the first time, that
Of course.
that um, the yeah, that the heat shock or heat stress on a copepod, like, in a northern
Mhm.
population that's not used to that, just like constitutively or just always having the heat shock proteins turned on, like
Right, right.
based on changes in the genetic blueprint level, they're able to like adapt more quickly, like up and down. Like if it gets cold, they're
Yes.
okay. If it gets hot, they're okay. Versus
Yes.
ones in the south, they kind of have adapted genetically over long periods of time to just like always have, some baseline of this. But you were looking at like, does that actually make them less flexible?
Yeah,
Which I thought
I
was
think
really interesting.
you're pretty spot on. I
Okay,
think you've got it.
okay.
That
Okay,
was a
okay.
good summary. But yeah, I think it's we're thinking about this a lot in terms of climate change, right? So when you're thinking about it getting warmer, that's a classic question to ask. And you might think that like it would make intuitive sense that those southern populations that are more warm adapted are going to do better in the
Right.
future, right? They're already ready to go. But what we're finding is that, they're ready to go at their current temperature, but if you keep increasing it, they're kind of at a ceiling where they might have less flexibility and ability to adjust. Versus in the north, their overall tolerance is lower, but we see that they have this higher plasticity, this flexibility. And so, really, as long as the temperature, increases at a rate that's not too fast, or there's lots of different things. There's other stressors and many. This is a prediction kind of in a scientific vacuum, but the prediction is really that in the north it's cooler, it'll keep getting warmer, and they should be able to adapt a bit better than in the south where it's hotter now but will get even hotter. They might be reaching kind of this critical moment where they might not be able to adjust any further because they're already doing everything they can right
Yeah.
now.
Yeah.
Yeah.
It's super interesting. And when I think about it, so we are both
Yeah, yeah.
in Louisiana.
It's hot.
It's very hot here. It's about to get very hot this summer. So me and
Yes.
in Louisiana, we get used to being warm. I mean, I know that like I have some family in Utah. I go there and it's like barely, 60 degrees and I'm freezing to death.
Yeah, yeah, totally.
It's just something, the plasticity is something that also happens in humans.
Yeah,
Yeah.
absolutely. I think about when I came to Louisiana, I had been working in Northern California for 6 years at that point doing my dissertation. And the first summer I moved here is when I started this experiment. And I was like, oh my gosh, I'm doing it. Like I'm living it with the
Yeah.
copepods
Yeah.
that first summer. And it was, 3 years ago at this point. That was the summer, if folks remember, in Louisiana was a heat wave and was a drought and like was incredibly hot, like for Louisiana standards. And I was like, what did I do?
Seriously.
Um, but that, I'm in my third summer. It's still really hot. I don't think I'll ever get used to it, to be totally honest. But I do feel like I've acclimated. Like I know. So think about plastic things we can do. I know, like I should not go for a run at noon on a
Right.
90-degree day,
Right.
right? Like I know that I got to get to air conditioning and I need to hydrate better. Like there are things like behaviorally and then physiologically we're doing to make those adjustments so that it doesn't feel quite as intensely stressful. Um, and things like heat shock proteins that we see in our copepods are really highly conserved. So humans have heat shock proteins, bacteria have heat shock proteins, plants. So that's a really common way for things to adjust and make sure everything in the cell, all the proteins in the cell are not misfolding or melting, basically. It keeps everything in order. But that's a mechanism that are in humans too.
Yeah, that's super cool. So what are you
Yeah.
doing now in terms of like with the centrifuging? You're looking to separate or, yeah.
Yeah. So we're at this phase. We just added some ethanol, like the, ethanol you'd find in your alcoholic beverage of choice. So really simple, a lab grade version. And that kind of mixes in with the RNA. And again, I can't tell you exactly what's happening, but does something to the chemistry that prepares it for our next step, which is this is just like it's a wash buffer. It's a proprietary recipe, so I don't know what's in it.
Gotcha.
But what we're gonna do here, I'll pull out our tubes. Um, so what you'll see here is we're gonna do this a few different times, but you've got this kind of collecting tube on the bottom, and then this column that fits in. And this column for the rest of the steps we're about to do holds on to our RNA. Like our RNA is not going to go anywhere. So it's just situated on this little filter, kind of right here. And then we're going to wash it with all these buffers that are going to get rid of anything that's not RNA that we don't want. So if there's any extra
Gotcha.
salts, if there's any extra DNA, if there's any extra, I don't know what else might be in
Yeah,
there.
yeah. Well, yeah, because I can see that
Um,
it's not red anymore. You don't have any of that
yeah,
like original red liquid you had in there.
exactly.
Nice.
And so everything that's not RNA is going to flow through if that centrifuge is spinning it through the column, and it's going to collect in that collecting tube. And what you'll see over and over is that I'm going to take that collecting tube, dump out what we don't want, reuse it, and then we'll put in the next thing. And we're just washing that filter, and it'll happen like 3 or 4 times. And then eventually, when we're ready, we'll put it's just water. It's like, very clean. We call it RNase free water. But at the very end, really, we're just going to put some water. We're going to let it sit on that filter for 5 minutes and then centrifuge it one more time. And our RNA will elute, is the fancy word, but it'll come out with the water. And that water in the tube will be what you send to the sequencer or do your downstream tests on. So it's
Gotcha.
it's like not anything crazy. It's ethanol and some, I think, just salty water, basically. I don't know exactly what's in there, but they're not crazy chemicals or anything. It's just something about that chemistry works. So yeah, I think it's like a little bit magical.
That's
I don't
amazing.
question it too
Yeah.
much.
So you're doing things like you taking a copepod, maybe putting in a heat bath, then you like flash make it cold again, right? Like flash freeze it to like get
Yes.
its genetic
So
material out, and then you see like at that point that it came out of the bath or was in the bath, like what genes was it expressing, right? Like,
exactly. And so that's I think the one thing, if I've learned anything about RNA, is that it's really unstable. It likes to deteriorate almost immediately. So DNA is a little more stable. You can like keep it on the counter, you can keep it in a fridge. RNA is just, it does not like being at room temperature. It does not like being out of its cell. And so we flash freeze the animals, like we'll pull them out of the hot water bath or whenever we're ready. And we actually snap freeze them in liquid nitrogen. So we like dump them in and they freeze immediately. And what that does is it preserves all the RNA that's in the cells at that time.
gotcha.
Like it's just hitting pause. And then when we're doing these extractions, so we missed the first little bit partially because it is me running around kind of like a chicken with its head cut off because you have to act really quickly. You've got to like blend the copepods and get them into buffer and get those first few
Gotcha.
steps started because as soon as they leave the freezer, it's going to degrade immediately. And what you'll notice, I actually have like a little cooler with ice for our last step because as soon as the RNA leaves these filters and is in water, it becomes unstable again. And so they're going to get popped into the ice and then immediately into our -80 freezer where it will remain until we use it.
Very cool.
So it's a little persnickety, the RNA.
Yes, yes, yes. So I've heard. Well, yeah, I think that's really interesting. I didn't know that there was this trade-off you mentioned where, like, if those southern populations that get kind of, um, potentially, if they're like, within their blueprint to maybe to express, I don't know, a particular protein or
Mhm.
something, but they're doing it kind of all the time, there
Yeah,
becomes a trade-off with, like, in the future, maybe they're not as flexible now to turn that back off, or maybe it's, like, metabolically expensive for them or, it
yeah,
has some kind of trade-off in terms of survival, and I hadn't
yeah,
thought about that before.
yeah. I think
Talked
if nothing else, like an ecologist, an evolutionary biologist, like the thing we look for is trade-offs because there's
Gosh,
always an interesting story when there's a trade-off. And almost everything in life isn't free, right? That's the general rule. So yes, making things like heat shock proteins, for example, are really energetically costly. So there's a lot of metabolic need involved. So you can imagine like yeah, maybe it's helpful to just flip that switch and keep it on. But just like keeping your light bulbs on, your electricity on,
Mm,
like there's a cost involved. And sometimes it's worth it. Maybe you can't not do that, right? Maybe you die if the light switch goes off. And so you just have to figure out how to cope and eat more or be less active or
mm,
have less babies, right? Those are often the trade-offs we look for. Um, just to be able to survive versus if you're able to turn the switch on and off when you're in the room, you're ultimately going to save on energy, but there might be a cost to flipping the switch, which I think is it's a little hard to separate that sometimes. And it's something that we as biologists who are studying plasticity have had a really hard time quantifying actually. Like, what is the cost of being flexible? Not just in terms of the trade-offs, but like how do you maintain a light switch, for example, basically? Like how do you allow things to go up and down? And is what does that cost? Because it can't be free, because otherwise everything would do that, right? Like if you could be perfectly plastic and just sense your environment and immediately do exactly what you needed to do, why wouldn't every single organism in every population just do that, right?
right. Yeah, that's what I was going to ask you. Like, when is
Yeah,
plasticity a positive and when is it a negative, right? And how do you figure that
that's the
out?
question.
And because I know
That
you've
is,
been studying also
yeah,
like copepods, maybe a mother copepod that's pregnant gets exposed to some stress, and then maybe the baby later actually is more potentially, changed for that reason, maybe more or less able to deal with stuff. Which I think is really interesting. But it's like, yeah, like you said, if we could all, if organisms could all do this, why don't we all do it?
yeah.
Yeah.
So, I mean, I think that's one of like my big questions of my scientific career, to be honest. So we know that plasticity can be adaptive, right? It can make sense to be flexible. It can save you energy. It can allow you to be, go to different environments that are maybe heterogeneous or varied. And so, that's great. But we also know it can be maladaptive. And so
Mm,
you can, one of the classic examples of like if you mismatch your cues. So if you think it's going to be hot and you turn on all these things and it turns out to be cold because you were wrong for whatever reason, that takes up energy that you didn't need to use. And so, um, different contexts, different ecological contexts will make plasticity either adaptive or not. And what we know from, more theoretical work and kind of mathematical modeling work, which I've been lucky enough to dabble with and people who know better than me, but what we see is that there's kind of a sweet spot. You want an environment that varies. Like if it stays the same, there's no reason to adjust, right? You should
gotcha.
just
Right.
turn the switch on and keep it at that level. Um, but if it varies, it might be worth being plastic. And if it's not too costly, right? Like if it doesn't cost you too much. And then the other piece is that it has to be somewhat predictable, right? Like if you're like the classic is plants over winter, for example, if there's a early melt and they start to bloom and then there's a freeze, they're all going to
Right.
die. They're not going to have babies. And so you need to be able to sense the environment reliably. You need to be able, and that environment has to predict the future environment. So that's, kind of the rules of plasticity as we understand it now. And then if you think about the scale over which that happens, so one of the things we looked at, or I've looked at, and in a paper that's coming out soon, hopefully, there's plasticity that can happen within a generation. That's kind of our classic understanding of it. So, like, for example, in the paper we've talked about, we looked at baby larvae who were exposed and saw how that progressed into adulthood. And sometimes it was helpful. So, in that northern population, the sensitive, it was good and adaptive. And then in the southern, it didn't really help all
Mm,
that much. I also am really interested in this concept of transgenerational plasticity, so parental effects. And what we find is if in this same northern population, if your parents are exposed to a heat stress, it actually carries forward in a negative way. And so you're like already at a disadvantage being
interesting.
born from stressed parents. So that's a case where it's maladaptive. In other cases, it's adaptive, and your parents can prime you and you do better because they're kind of gifting you this information from their environment to change your phenotype. And so this is this concept of non-genetic inheritance where you can inherit, a phenotype, but it has nothing to do with your DNA. It's, we're still figuring out mechanisms, but
Right. Right.
epigenetics is one we think about a lot. Maternal provisioning, so like how much nutrients are in the eggs that mom makes. That's another common. I think that may be what's happening in our copepods potentially. So if the mobs are heat stressed, they have less energy to waste or to use. And so they might be making lower quality eggs, for example. And so when those larvae hatch, they're at a disadvantage because they already are in a deficit energetically.
Uh,
So yeah, plasticity
yeah.
can, it's awesome, but it gets complicated.
Yeah, yeah. I mean, so in terms of studying these complicated things, I mean, what's
Mhm.
been I don't know, when did you think you were gonna end up studying plasticity? Like, how did you fall into that? And then how do
Yeah.
you imagine that's yeah, gonna turn into impacts? Like, what do you hope that your research is gonna do for for the
Yeah.
world? I mean, I'm sure you are out there in the field, like even in California, right? Like collecting
Yeah.
copepods, you see the environment. I'm sure that you, just like the rest of us, like see changes and have
Yeah.
struggles with the
Yeah.
accepting that. So like, yeah, how do
Yeah.
you?
Yeah. Okay, I'll try to think where to start. I guess I'll start kind of backwards and answer your most recent question, which is thinking about and we talk about this a lot when you study climate change, like how do you stay optimistic? It can be
Right,
very doom and gloom,
right,
right? And I see that in my studies. I have some where it's just like, okay, like heat stress is really bad for you, or ocean acidification is really bad for you. And the more you get exposed, the worse it is. Like that plasticity just compounds and is negative.
gotcha.
Um, and I think when we understand that and can more accurately predict what will happen at a species level, at a population level, it's going to help us better understand conservation. Where who do we target? So, like, we thought southern population is logical. You'd think maybe that's the one to keep around because it's
Right,
got this resilient genotype. But really, maybe it's the northern population. And if you're gonna, assisted gene flow, for example, that's a little outside of my scope, but that's something we think about. Um, if you're going to do conservation, like can you move copepods around, for example, and bring those beneficial genes to where they need to go so that populations can persist? Where is the need greatest? And coastal California, that's where I did my PhD. It's also where I did my undergrad, so it's a very special place. And I'll just, it's an El Nino year, if you've seen that in the
right,
news. It's been a lot of
yes,
headlines.
yeah,
So we're expecting the water to get really warm this year. And the last time when I was an undergrad working out at the coast, we had our first El Nino warm blob heat wave, and I watched everything change. So the kelp all died off, the sea stars died off, and that was due to a disease and some other factors as well. But there were some big changes between 2013 and like 2016, and that was right as I was finishing my undergraduate degree and getting interested in ecology and evolution and marine biology. And so it definitely had an impact. And then I actually went kind of on a tangent and was a research technician in the coast of North Carolina for a couple years after I graduated. And I studied hurricane damage on the coastline and kind of more of an applied question, and really thinking about how our weather pattern changes, how a sea level rise. Um, going to impact homeowners and marsh and natural habitats, and how can we think of these kind of hybrid solutions that marry ecology with engineered structures to keep the shoreline where we want it as humans, but also functional for all the fish and crabs and everything that call it home. So then I guess this circles back perfectly to kind of the first part of your question. um, so I was really kind of on this applied track, but the questions I got to ask as an undergrad, I was so privileged. I did an honors thesis and worked actually with plants. Um, I studied transgenerational plasticity, and that was my introduction to biology and evolutionary ecology. And I never forgot those questions. They never stopped being interesting to me. And so, even though I kind of changed paths and became a marine ecologist, I decided in my PhD to marry those two. And that's kind of where my work comes from, getting to ask the intellectual questions I'm interested in marine systems, which I find particularly exciting. And I always, in the back of my mind, try to think, what is the applied part here? And
right,
that's not quite what my work does anymore. I'm a little more of the basic science side of things. But thinking about conservation, thinking about aquaculture is another big place we can apply these questions. So in my PhD work, I worked with abalone, which are these like big snails that Californians really like to eat. That's like a very culturally important species that is endangered now due to temperature rises, among other
Wow.
things. And so I studied the impacts of ocean acidification, which is another climate change stressor, um, in the context of how can we buffer critical life stages to maximize aquaculture output? How can we understand plasticity and how stress carries forward, from larvae to adults and then parents to offspring? And can we find these little windows? Because they're long lived. The copepods are short lived. The abalone are like 30 plus years.
Oh, wow.
So how can we, like, if we, what we found is that if you keep the pH at a good level in the larval phase, which is like 7 to 10 days, it carries forward positively for the rest of their life. And that's hard to do in the ocean. You can't go and like put baking soda in and keep it basic,
Wow.
But if you're growing them, you can. And then you can potentially release them and do reintroductions. And there's a conservation lens too. So
Oh, interesting.
that's kind of the mismatch of my work. It's just a little all over the place, but the theme is actually plasticity, weirdly enough.
Yeah, no, that's super interesting. Yeah, so like
Yeah.
by studying like what you could do in that, like you said, short amount of time, I mean 7 days, if you could do something there that has outsized effects for its survival for the rest of those 30 years, that would be where you focus your efforts.
Yeah.
Which I think that, you know, it just occurs to me, like a lot of scientists working on things. I don't know. I could be wrong, but I feel like a lot of people when we think about science, we're like, oh, scientists solving problems, which it's not always. I think maybe sometimes we get frustrated as scientists because we're not always
Yeah.
solving the
Totally.
problem,
No,
but like,
absolutely not. Yeah.
but being able to, I just identify the scope of the problem and like
Yeah.
where is it best to focus the efforts. I know, like in the pharma
Yeah.
industry, like there's been a lot of like abandoned discovery efforts because they need to
Yeah.
kind of efficiently use their money, and if they don't know where
Right.
to focus, like they're just like, oh, we're just kind of giving up on this, it costs too much money, versus scientists can be like, this is, point to one direction and say this is the most
Yeah.
likely to work, then like the funding can be more effective like in that
Yeah.
one particular direction. So I think maybe in this case it's like that, like maybe you
Yeah.
can't solve the whole problem, or like there's still going to be climate change, there's still going to be negative effects, but figuring out where's like the most bang for our buck to make a difference.
Yeah, I mean, this is like maybe slightly more doom and gloom side of things, but the analogy I've heard is it's like triaging, right?
Right,
Like
right.
everything's kind of in trouble. Our planet is changing and it's changing rapidly and it's changing, there are some winners and some losers, right? It's unevenly distributed, but we need to figure out who's being hurt the most and um you know who can we help, like what's a reasonable place to, like we only have so much effort and money and things like that, but also I think the lens of ecology is really helpful for that too, because the species that the pandas are really cute, but like ultimately it's like the
Right.
nematodes that are gonna, we need bees, we need little insects, we need microbes, the community is important and that holistic picture is really important. And so yeah, I think there's always kind of a nice way to tie back, but it's not always apparent when you read a paper. It's not always apparent, when you're pipetting for millions of hours. Like it's
Yes.
helpful to kind of zoom back out and be like, yeah, why am I doing this?
Well, speaking of that, that's a great segue because I was going to ask,
Yeah,
shift gears a little bit of like, what's, life for you as a scientist like? I
yeah,
mean,
yeah,
what do you really enjoy about it? Maybe what are the struggles? Are there any struggles in this particular moment, especially for you, or?
yeah. Yeah. Um, yeah, okay, so day in the life. I mean, the thing I really like about being a scientist, there's many things, but one of my favorites is that it varies. Like day
Yeah.
to day is different. Like I love that variety. And I love that the science kind of carries you to what that looks like. Lately in my postdoc, again, I've been doing a lot of molecular work, so lately it's been a lot of extractions. Um, I just finished a lot of those. I'm writing up papers. So, the culmination of several years of work is finally kind of coming together in a series of papers. I've been doing a lot of writing lately, which means a lot of computer time,
Yes.
unfortunately.
Yeah.
Data analysis. So that's been kind of my day to day. So sometimes that means I'm coming in and I'm 8 hours at the lab bench just trying to get through my RNA extractions. Sometimes that means I go tuck away in the student union or somewhere quiet and I try and write for the day. Um, and so that's what it's been lately. And I'm hoping sometime soon, because now we're wrapping things up in the future. The thing I miss the most is field work. I
I was
love
gonna
field
ask,
work. I
yeah,
love
like,
being outside.
yeah. So I mean, you have some
Um,
great pictures I'll share of like
yeah, yeah,
being in these where we didn't discuss where you find the copepods, but in these like splash
yeah, yeah,
pools where like the water kind of
yeah,
splashes onto rocks and it forms a little puddle, and then it can, yeah, the copepods are in there, and that's why they can experience ranging temperatures. But you have some great pictures of being kind of,
yeah,
the ocean enveloping you and going
yeah.
and looking at these tide pools and collecting buckets of copepods. What do you miss most of it, field work? And are you hoping to get back to it? Is that something you will do?
Definitely hoping. Yeah, I think so. And that was a lot of my dissertation work because sorry, I'm going to set a timer quickly. We're almost done.
Yay!
I came to my postdoc with the goal of learning these more molecular skills, and I've learned them in spades. It's been wonderful. I love learning new things, so zero complaints. Because I came from a background of more manipulative experiments and field work. So more observation, going out in the field, taking field measurements, and then bringing animals back to the lab and manipulating their environment in a lab setting and taking lots of data and keeping all those animals alive for many, many months. And um, I like, again, I like the variety,
Yeah.
but I'm definitely in my cycle of ready to be back. And like pulled copepods for these extractions today. And I was like, I haven't like looked at a live animal in too long. Like, I have a
Oh no!
dog and I go outside, but like getting my hands wet, literally,
Yeah.
getting into the salt water, even if it's in the lab setting. So what's nice is, members of my lab are working with like periwinkle snails and they do marsh work. And so I can kind of live vicariously and help them as a field hand from time to time. So that's how I've been getting my fix lately.
Oh, nice.
Um, but yeah, I think I like the kind of trajectory I took in that. I think like the natural history to me is so, so important. Like the working with these copepods, I've learned so much. Like the science questions I get to ask, my intellectual curiosity is really satisfied, and I'm learning all these skills and different ways to look at things. And it's been amazing, but I think is underlined by this, like, I've been to these tide pools, I've seen these animals in the wild. I know, like, when we see population level differences, I can be like, well, it makes sense. Like, the rocks are different colors, or like, well, that
Oh,
pool
yeah.
was really deep, like that. I think sometimes, not to call anyone out, but sometimes when you're in a lab where you're doing this kind of bench work, I don't think you always know even like the wild version of the organism you're working with. And that's
Right.
fine. Everyone needs to do their part and there's value in all of it. But my passion starts with nature and seeing how they live in their environment and what they're up to and what their behavior is. And then we can link that to all the kind of heavy science.
Yeah, no, I think that's super interesting. I was gonna say, because I
Yeah.
think that there are, right, what scientists call model organisms, or I guess the
Yeah. Yeah.
organisms that like, mm, science is just kind of, well, actually, you could probably know it better than me, but
Sure.
I guess organisms that people use commonly that have become maybe standardized, where you can use
Yes.
them in one lab and another and kind of be able to make
Mhm.
comparisons. I don't Your copepods you're working with are not model organisms, right? So
No.
you've had to go kind of get them and figure out how to
Yeah.
reproduce them, not reproduce them, but how to take care of
Yeah.
them in
Yeah.
the lab and
And,
standardize them in some kind of way.
to be totally clear, I'm sitting on the shoulder of giants. There
Yeah.
are people here who have figured that out. So I came into a new system for me, but there was a lot of information. So I just want to
Gotcha.
fully acknowledge the
Gotcha.
collaborative nature of science.
Gotcha. Yes.
Um, but I think it's a really good time for people who are interested in molecular work and molecular questions for non-model systems, because our toolkits are just becoming so much bigger, and it's cheaper, right? Like we can make genomes, reference genomes in a way that we couldn't 10 years ago, for example. Like it just cost so much money and was prohibitive. We actually, the copepods I'm working with now are Californicus, so the California coast. They go all the way to Alaska to Baja California, which is wild. But I'm also working with their South American congener. So that same Tigriopus genus, but the Angulatus. So they're in South America along Chile and Peru. So we have some collaborators collecting the samples for us now. Fingers crossed they'll come back
Wow.
okay. Um, and so I was able to assemble that genome so we can ask the
Wow.
same questions we're asking here in a parallel gradient, and see if the patterns we see in North America are the same in South America because that latitudinal range is, the same, just south and north. Um, but like that would have been, it was expensive. We had a nice grant that helped us fund that, but I think that wouldn't have been possible, you know,
No,
even,
it's true, yeah. I've heard,
yeah.
yeah, talking to scientists, and that's something that's kind of in the weeds, but it's so
Mhm.
true. I mean, the rise of technology for
Mhm.
sequencing, even AI,
Yeah,
for
yeah.
and data science for kind of, pulling that data
Mm.
together and making sense of it, that now we're able to do genome sequencing on non-model organisms, which just means, like, I guess more organisms that are you're pulling maybe from the wild that haven't been kind of like bred in the lab for a very specific reason, where in
Yeah.
the past maybe people had to more so work on those because they didn't have the tools or the money to go just pick a new organism and sequence its genome and figure out all these things that
Yeah.
you're figuring out now.
Absolutely. And I think I study marine invertebrates, which are often underappreciated in my
Yes.
humble opinion. And I think, the a huge portion of our biological diversity is in the ocean and is marine invertebrates. There are so many phyla. There's, at least one or two that are only found in the ocean. You think of all the diversity and the evolutionary histories, it's just insane in the ocean. And so if you really want to understand patterns of life, like you've got to, sample that breadth. And so, we know how things work in fruit flies. That doesn't mean that it's the same for
Right.
our copepods. And that's the closest model system to our copepods.
Really?
And they are super diverged. So I'm trying to remember the specific, but it's like as far away as humans from mice, at
Really?
least, if not more. They're both arthropods, they're in the same phylum, but they are really nothing alike. So we use as much as we can because there's so much more tools and information and things they're able to do with mice and fruit flies and everything else is
Right.
incredible. Um, but like it's actually not close at all.
Wow. Oh, yeah, I didn't even
Yeah.
think about it
Yeah,
that
yeah.
way. That's super cool. Well, are there things you wish more people knew about science, about the process of doing science, about your
Ooh,
science in your field?
yeah. I'm trying to think. I think it feels like this has just been on my mind a lot lately, given our current science discourse, I would
Right.
say, in the broader, political and otherwise. It's an iterative process, it's an exploratory process, it's question driven. I think when you're in school and you're studying a textbook, it feels very much like we know how the world works and like you're being handed down this information from above and this is the way it is and has always been. And it's just completely not true. And we disprove things all the time and we learn new things all the time and we walk back things we thought we knew and we challenge things. And that's a normal part of science. That means it's functioning well. But I think it's scary when you don't know, well, that you think a drug is effective and it turns out there's side effects, or we say that something is healthy for you and it turns out it causes
Right.
cancer. Like I understand why there's this that can lead to this feeling of, distrust or misunderstanding. And I think just knowing that, like, yeah, that being able to engage with some of that literature, even if you're not reading the papers themselves, but thinking critically as you read a news article that like this is probably only part of the story. And like,
Right.
there's going to be caveats and nuance. And unless there's multiple studies that show it, I wouldn't take it as fact just yet.
Right.
Um, so, when you do it day in and day out and you see for yourself the hypothesis you put forth is definitely like not what happened.
Right. That's true,
But
yeah, I guess, yeah. I mean, I think it's how our human psychology works, honestly, like, to
totally. Yeah.
simplify, and it's something even scientists, we have to work through, you know?
Yeah.
But like you said, I'm more like probably just living it and having that be getting in the habit of knowing that what you know now, doesn't mean it's gonna be like just
Mhm.
a linear building process on that knowledge.
Right. And, the flip side of that, and speaking to someone who thinks about science communication, it's something I think about. It's why I was excited to chat with you today. We as scientists can be much better too, because we're trained to be in this language of caveat and complexity, and we don't say things very forcefully because we're trying to be very careful. And I think that it's like part of the process, but also makes it really hard to, have a conversation with lay people or someone who doesn't know your field. Um, and just figuring out how to simplify and make sure our message is getting across, faithfully and like representing the data as it is, but in a way that's more accessible because it's so inaccessible as it
Yeah.
stands. The way we publish papers, the way we write our papers, the way we talk to each other about our papers is
Yeah.
very jargon heavy, and it doesn't have to be that complicated. So I think it's a skill and a muscle that we should all be strengthening and flexing. And I think as a community, we're getting better and better, but there's definitely a long way to go. So
Yeah.
it's something I think about.
That's awesome. Well, Isabel, thank you so much for chatting with me today. And I
Yeah.
know, what are you wrapping up now in terms of putting this stuff on
Yeah, here,
ice?
I'll show you the very exciting final product. It's just
Woohoo!
water.
It looks very clear.
So yeah, that's, we'll go and do all kinds of things with this.
That's awesome. So that's just, yeah, pure RNA that you've
It's
extracted
just
from. Yeah.
RNA in water.
Nice. Nice.
Yeah. Yeah.
And you said you send that off for like RNA sequencing with a service or company outside of LSU.
Yeah, so that's the work we've been doing. We go and we get the whole, it's called the transcriptome, right? So
Gotcha.
all this RNA gets sequenced, and all those transcripts we compile and are able to, we map it to the genome. So again, RNA talks to DNA. And it's just like really powerful way to look at RNA, but there's definitely other ways to do it. There's simpler, more of the cheaper ways too. But for this study, we went all in. So yeah, we
That's
send it off
awesome.
and have someone else do it.
Well, I mean, that also means, right, you don't necessarily know what you're looking for. You're just looking at
Mhm.
like all RNA that's being expressed,
Yep.
not like, I guess it would be cheaper, or if you have a specific gene, you're like, well, I want to know if
Yep.
that's on or off. You could just look for like just a signature of that piece of RNA. But
Absolutely. Yep.
transcriptomics is much broader, where you're kind of saying, well, I don't even know necessarily what I'm looking for. I know what I'm looking
Yep.
for, but I think modern
Give me the whole
science,
thing.
right? I
Yeah.
think modern
Yep.
science, which I think is super cool, is almost like give me everything, and
Yeah.
then I will like use machine learning and data science and all that to like start to make sense of that, which I guess in the
Yeah.
past very much we haven't done in that kind of same way, right? Because it was
Yeah.
like that's too
We just
much.
couldn't.
Yeah,
Yeah.
it couldn't. And so then it'd be like you had to focus. And I think sometimes even now there's in medicine, there's still that shift of like if you test everything, you're gonna find everything, but now it's
Yeah.
like,
Yeah.
no, our tests are getting better to where like you can start to like test for things, just because. Yeah, interesting.
Yeah. Yeah. Totally.
Awesome. Well, thank you so much.
Yeah,
And
of course.
yeah,
It was, always a pleasure to talk about my science and have someone listen.
sweet.
My name is Isabelle Neylan. I'm a postdoctoral research fellow. I'm funded by an NSF postdoctoral research fellowship in biology, and I work at Louisiana State University.